Hello, and welcome to another screencast where I'll be discussing muscle physiology 1B. This is going to be the excitation contraction coupling part. So before you embark on this, you should understand and review the microanatomy of the muscle fiber. You should already know what the sarcomere is, and then the detailed structure of actin. That is the three parts of actin, of the actin molecule, and then the two parts of the myosin molecule. We're going to focus here on this sliding filament mechanism, and by mechanism we mean that this is the way we mean that this is how it works. All right? So the mechanism is how the muscle is actually contracting. It's going to involve calcium ions, the troponin, tropomyosin complex, actin and myosin, and we're going to involve ATP here. And this is also sometimes known as the cross-bridge cycling mechanism here. So let's get started. So here again is the sarcomere. Again, the sarcomere goes from Z disc to Z disc, and it contains the overlapping actin and myosin filaments here. We're going to pay close attention to this interaction between the myosin heads and the actin here for the rest of this. So again, just a quick review. You have the actin molecules here with the active sites that myosin heads will bind to. Here's the tropomyosin molecule that will cover those active sites. Here's your troponin molecule, again with the actin binding part, the calcium ion binding part, and then the tropomyosin binding part. Your myosin myofilament is a little bit thicker and consists of groups of the individual myosin molecules, and you have this rod portion here that is like the handles of a golf club, and then you have the myosin heads here with active or ATP binding sites on them. So again, we're going to focus on the interaction of those. And what happens in the cross bridge, a cross bridge is just this, where you get the myosin heads interacting with the actin molecule. So this is what we call the cross bridge. So the cross bridge is going to be where the myosin heads will bind to the actin molecules here. So a little bit of background. Right? This ATP molecule is an energy-rich molecule, and when you break one of the bonds there, you end up getting ADP, adenosine diphosphate, plus an inorganic phosphate. And this reaction here makes energy available. Right? So energy is available here to get this myosin molecule to bind to the actin and then cause some sort of reaction or contraction here. So basically what's happening is the sarcomere is going to change its size. Right? The actin molecules are going to be pulled across the myosin molecules. And so here we see the muscle relaxed, and as the muscle contracts, those actin molecules are going to be pulled over the myosin molecules. So here's the muscle completely contracted. And you'll notice that the H zone is completely gone right here. That is because the actins are slid over one another. Those myosin molecules, let's relax the muscle. So there it is, relaxing, and then there it is, completely relaxed and almost all the way stretched. So now you compare here, the H zone is much larger. So here's the H zone, and the H zone here, when the muscle was contracted, was completely gone. Notice that the actin and the myosin do not change their length. You know, the actin molecules are simply pulled over the myosin molecules. So that actin is not going to change its length. It just moves. The myosin does not change its length. It's just pulling the actin across it in that muscle contraction. Okay? There it is, much more static. But let's talk about the mechanism here a little bit more detail, going back to the action potential that's down the T-tubule. So in a previous screencast, we had addressed the neuromuscular junction here. So the action potential is going to come down the neuron here, acetylcholine is released into the cleft. That acetylcholine then will trigger the action potential to move down the sarcolemma, which is what the arrow is showing here. So as that action potential moves down the sarcolemma, it's then going to move down this T-tubule, and the action potential will trigger the release of calcium ion from the sarcoplasmic reticulum, from the terminal cisternae of the sarcoplasmic reticulum. That calcium ion here that's released will then bind to the troponin molecules. This is what we see happening down here. So calcium ions are going to bind to the troponin molecule that slides the tropomyosin off. This now allows an energized myosin head, shown here, to form a cross bridge, and that myosin will change its shape. The myosin molecule actually changes shape after it binds. I don't know what is wrong with this here today. Changes shape, and that change of the shape then pulls the actin in this direction and away from the Z line. That calcium ion, if it remains in there, that muscle will continue to contract. Right? That muscle will continue to contract, but the calcium is then removed from the sarcoplasm and or the cytoplasm, and then gets moved back into the sarcoplasmic reticulum. When that happens, the tropomyosin here slides back over the actin active sites, and the myosin head here is re-energized with a new ATP molecule, and then returns to its active conformation. So when you see the actin like this here, when you see the myosin like this, it is ready to be contracted, ready to be activated. Right? So that is the mechanism statically. I highly recommend running the PowerPoint, looking at the animations that are there. Looking at this thing in motion is much more effective. Again, here is that cross-bridge cycling. Again, we'll start at the top at about 12 o'clock here. You see that the myosin has been energized here with the ADP and inorganic phosphate attached to it. So this is energized, ready to go. You assume that calcium ion is already bonded to the troponin here in yellow. The tropomyosin, you notice, is slid off of the active sites of the actin. That then allows for cross-bridge formation. You get the cross-bridge formation. You get the myosin molecule changing its shape while it's still bound to the actin here, and it pulls the actin in this direction. That releases any ADP. Now that's going to free up a spot for a new ATP molecule here to come in and bind to the myosin head. When that new ATP binds to the myosin head, that drops the head, that breaks the cross bridge, and that ATP will be hydrolyzed or broken down to ADP and inorganic phosphate here, re-energizing the myosin head. And if calcium ion is still present, then it binds to the troponin, which slides the tropomyosin off, and the myosin head can then bind to the actin and then pull again. Right? And this will keep happening as long as calcium ion is present. When calcium ion goes away, then the tropomyosin slides back over the actin active sites. Again, 4 animations here that would recommend you taking a look at. 4 animations there, taking a look at that's going to put that all in motion for you. Okay. Again, just more static diagrams of the same, describing the whole cross-bridge cycling. So here, this ADP and inorganic phosphate, I've energized that myosin head, calcium ion present. Here we get the removal of the ADP and the inorganic phosphate. That cross bridge is then going to change its conformation right here. So we lose this ADP. That myosin head is bound to the actin. That is a pulling motion. Again, we're going to pull the actin in that direction. We get a new ATP coming in here, and when you get a new ATP coming in, the cross bridge is broken right here. You see that there's no connection, and then that myosin head can return to its energized state right here. So this is it energized, ready to go. and then bind again to the actin molecule. Okay, so that's going to pivot back there. All right, so hopefully you are able to describe here and explain the sliding filament mechanism. That's how the sarcomere actually is shortening, involving calcium ion, the tropin and tropomyosin, actin and myosin, and ATP there, and a little bit about the cross-bridge cycling. Okay, thanks for listening. Bring your questions to class.